Welcome back to Concepts of Biology. We'll continue to read Chapter 2, Section 2.3, Biological Molecules. By the end of this section, you will be able to describe the ways in which carbon is critical to life, explain the impact of slight changes in amino acids on organisms, describe the 4 main types of biological molecules, and understand the functions of the 4 major types of molecules. The large molecules necessary for life that are built from smaller organic molecules are called biological macromolecules. There are 4 major classes of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids, and each is an important component of the cell and performs a wide variety of functions. Combined, these molecules make up the majority of a cell's mass. Biological macromolecules are organic, meaning that they contain carbon. In addition, they may contain hydrogen, oxygen, nitrogen, phosphorus, sulfur, and additional minor elements. Carbon. It is often said that life is carbon-based. This means that carbon atoms, bonded to other carbon atoms or other elements, form the fundamental components of many, if not most, of the molecules found uniquely in living things. Other elements play important roles in biological molecules, but carbon certainly qualifies as the foundation element for molecules in living things. It is the bonding properties of carbon atoms that are responsible for its important role. Carbon bonding. Carbon contains 4 electrons in its outer shell. Therefore, it can form 4 covalent bonds with other atoms or molecules. The simplest organic carbon molecule is methane, in which 4 hydrogen atoms bond to a carbon atom. Figure 2.13. Carbon can form 4 covalent bonds to create an organic molecule. The simplest carbon molecule is methane, depicted here. However, structures that are more complex are made using carbon. Any of the hydrogen atoms can be replaced with another carbon atom covalently bonded to the first carbon atom. In this way, long and branching chains of carbon compounds can be made. The carbon atoms may bond with atoms of other elements such as nitrogen, oxygen, and phosphorus. The molecules may also form rings, which themselves can link with other rings. This diversity of molecular forms accounts for the diversity of functions of the biological macromolecules and is based to a large degree on the ability of carbon to form multiple bonds with itself and other atoms. Figure 2.14. These examples show 3 molecules found in living organisms that contain carbon atoms bonded in various ways to other carbon atoms and the atoms of other elements. A. This molecule of stearic acid has a long chain of carbon atoms. B. Glycine, a component of proteins, contains carbon, nitrogen, oxygen, and hydrogen atoms. C. Glucose, a sugar, has a ring of carbon atoms and 1 oxygen atom. Carbohydrates. Carbohydrates are macromolecules with which most consumers are somewhat familiar. To lose weight, some individuals adhere to low-carb diets. Athletes, in contrast, often carb load before important competitions to ensure that they have sufficient energy to compete at a high level. Carbohydrates are, in fact, an essential part of our diet. Grains, fruits, and vegetables are all natural sources of carbohydrates. Carbohydrates provide energy to the body, particularly through glucose, a simple sugar. Carbohydrates also have other important functions in humans, animals, and plants. Carbohydrates can be represented by the formula CH2O subscript N, where N is the number of carbon atoms in the molecule. In other words, the ratio of carbon to hydrogen to oxygen is 1 to 2 to 1 in carbohydrate molecules. Carbohydrates are classified into 3 subtypes: monosaccharides, disaccharides, and polysaccharides. Monosaccharides. Mono equals 1. Saccharide means sweet. Monosaccharides are simple sugars, the most common of which is glucose. In monosaccharides, the number of carbon atoms usually ranges from 3 to 6. Most monosaccharide names end with the suffix -ose. Depending on the number of carbon atoms in the sugar, they may be known as trioses, 3 carbon atoms, pentoses, 5 carbon atoms, and hexoses, 6 carbon atoms. Monosaccharides may exist as a linear chain or as ring-shaped molecules. In aqueous solutions, they are usually found in the ring form. The chemical formula for glucose is C6H12O6. In most living species, glucose is an important source of energy. During cellular respiration, energy is released from glucose, and that energy is used to help make adenosine triphosphate, ATP. Plants synthesize glucose using carbon dioxide and water by the process of photosynthesis, and the glucose, in turn, is used for the energy requirements of the plant. The excess synthesized glucose is often stored as starch that is broken down by other organisms that feed on plants. Galactose, part of lactose or milk sugar, and fructose, found in fruit, are other common monosaccharides. Although glucose, galactose, and fructose all have the same chemical formula, C6H12O6, they differ structurally and chemically and are known as isomers because of differing arrangements of atoms in the carbon chain. Figure 2.15. Glucose, galactose, and fructose are isomeric monosaccharides, meaning that they have the same chemical formula but slightly different structures. Disaccharides. Di means 2. Disaccharides form when 2 monosaccharides undergo a dehydration reaction, a reaction in which the removal of a water molecule occurs. During this process, the hydroxyl group of one monosaccharide combines with a hydrogen atom of another monosaccharide, releasing a molecule of water and forming a covalent bond between atoms in the 2 sugar molecules. Common disaccharides include lactose, maltose, and sucrose. Lactose is a disaccharide consisting of the monomers glucose and galactose. It is found naturally in milk. Maltose, or malt sugar, is a disaccharide formed from a dehydration reaction between 2 glucose molecules. The most common disaccharide is sucrose, or table sugar, which is composed of the monomers glucose and fructose. A long chain of monosaccharides linked by covalent bonds is known as a polysaccharide, poly meaning many. The chain may be branched or unbranched, and it may contain different types of monosaccharides. Polysaccharides may be very large molecules. Starch, glycogen, cellulose, and chitin are examples of polysaccharides. Starch is a stored form of sugars in plants and is made up of amylose and amylopectin, both polymers of glucose. Plants are able to synthesize glucose, and the excess glucose is stored as starch in different plant parts, including roots and seeds. The starch that is consumed by animals is broken down into smaller molecules, such as glucose. The cells can then absorb the glucose. Glycogen is a stored form of glucose in humans and other vertebrates and is made up of monomers of glucose. Glycogen is the animal equivalent of starch and is a highly branched molecule usually stored in liver and muscle cells. Whenever glucose levels decrease, glycogen is broken down to release glucose. Cellulose is one of the most abundant natural biopolymers. The cell walls of plants are mostly made of cellulose, which provides structural support to the cell. Wood and paper are mostly cellulosic in nature. Cellulose is made up of glucose monomers that are linked by bonds between particular carbon atoms in the glucose molecule. Every other glucose monomer in cellulose is flipped over and packed tightly as extended long chains. This gives cellulose its rigidity and high tensile strength, which is so important to plant cells. Cellulose passing through our digestive system is called dietary fiber. While the glucose-to-glucose bonds in cellulose cannot be broken down by human digestive enzymes, herbivores such as cows, buffaloes, and horses are able to digest grass that is rich in cellulose and use it as a food source. In these animals, certain species of bacteria reside in the rumen, part of the digestive system of herbivores, and secrete the enzyme cellulase. The appendix also contains bacteria that break down cellulose, giving it an important role in the digestive systems of ruminants. Cellulases can break down cellulose into glucose monomers that can be used as an energy source by the animal. Carbohydrates serve other functions in different animals. Arthropods, such as insects, spiders, and crabs, have an outer skeleton called the exoskeleton, which protects their internal body parts. This exoskeleton is made of the biological macromolecule chitin, which is a nitrogenous carbohydrate. It is made of repeating units of a modified sugar containing nitrogen. Thus, through differences in molecular structure, carbohydrates are able to serve the very different functions of energy storage, starch and glycogen, and structural support and protection, cellulose and chitin. Figure 2.16. Although their structures and functions differ, all polysaccharide carbohydrates are made up of monosaccharides and have the chemical formula CH2O subscript N. Registered Dietitian. Obesity is a worldwide health concern, and many diseases such as diabetes and heart disease are becoming more prevalent because of obesity. This is one of the reasons registered dietitians are increasingly sought after for advice. Registered dietitians can plan food and nutrition programs for individuals in various settings. They often work with patients in healthcare facilities, designing nutrition plans to prevent and treat diseases. For example, dietitians can treat a patient with diabetes how to manage blood sugar levels by eating the correct types and amounts of carbohydrates. Dietitians may also work in nursing homes, schools, and private practices. To become a registered dietitian, one needs to earn at least a bachelor's degree in dietetics, nutrition, food technology, or a related field. In addition, registered dietitians must complete a supervised internship program and pass a national exam. Those who pursue careers in dietetics take courses in nutrition, chemistry, biochemistry, biology, microbiology, and human physiology. Dietitians must become experts in the chemistry and functions of food, proteins, carbohydrates, and fats. In our next section, we will go to lipids.